Welcome to CHM5403 Sensor Technology. For this topic, we will discuss about immunosensor. Before we can proceed, we have to discuss about antibody. So this is the structure of antibody. This is a amine group where antigen will bind here. This is the carboxyl group site of the antibody structure, and this will be attached to the transducer. Antibody will bind to antigen, and antigen are parts of the probably virus, bacteria, or any other pathogen. Antibody does not bind to the whole pathogen, virus, or bacteria, but instead they bind to certain epitopes of the antigens, and this is what the difference between antigen and antibody. The selective interaction between antibody and antigen is the basis of immunosensor. The interaction can be detected through optical or electrochemical as well as any other transducer technique. Antibodies can be divided into polyclonal as well as monoclonal. So for polyclonal, the binding site is quite general. So there are many epitopes on top of the antigen is available or can be bound with polyclonal. But monoclonal only bind with specific epitope on top of the antigen or surrounding the antigen. For the purpose of biosensor, the use of monoclonal is much more preferred. Let's say if we are talking about antigen test kit for SARS-CoV-2 or COVID-19 disease. So as you can see here, there are a few proteins available for detection. For example, spike glycoprotein or S protein, nucleoprotein, N protein, as well as E protein. These are the antigen that reacts to the antibody designed in order to produce the antigen test kit. So each of these antigen site or the protein requires different set of antibody. What are the differences between molecular tests or PCR tests, antibody tests, and antigen tests? So PCR tests or molecular tests is based on RNA or DNA. And for the purpose of detecting viruses, for example, SARS-CoV-2, molecular test or PCR test will be based on the real-time current infection. It will detect the virus that is in the sample from nose or throat swab. Same like the antigen test, where it also detects the antigens. For example, just now, the S protein, N protein, and E protein of the SARS-CoV-2 virus. And it is a real-time test where the virus is detected in nose swab or throat swab sample. But for antibody tests, it tested the antibody response of the infected patient or infected people. And it only indicates that the person has been infected before. So antibody test is not useful for detecting the COVID-19 infection, but probably it is useful if for the future we are to study the prevalence of the COVID-19 in certain areas. Definitely, PCR test is much more sensitive compared to the antigen test. This is an example of immunosensor where we use ELISA technique. Okay, as you can see on the surface of the transducer or the sensor, we can build or attach a capture DNA,
capture
where the purpose
antibody.
of capture DNA is to catch the antigen or any target molecule that pass through here. And we have another set of antibody which attach to a marker. The marker can be enzyme, can be also nanoparticles, or any other suitable markers for electrochemical, optical techniques. This is an example if we are using gold nanoparticle with combination of enzyme, where we can observe different of color between positive and negative reading. This is an example of the previous technique being applied on real sample. As you can see, sample from the positive TB patient appeared to be dark blue, whereas sample from negative TB appeared to be red. Another common example of immunosensor is based on lateral flow. This is quite common, where people usually refer as test strips. So in here we have the analytes. It might be serum, it might be sputum, or any, for example, throat swab or nose swab. And if the virus occur in that sample, so the specific antibody design to bind with the antigen from the sample will bind with the antigen, and it flows here and indicated on the test line. This is an example of the usage of lateral flow on TB positive sample. You have here the two lines, okay, of the test line and the control lines, okay, 1 and 2. And as you can see, for a negative sample, there is no line for the test line. Immunosensor can also be developed using electrochemical technique. As you can see here, the same principle, the same ELISA principle can be applied. We have capture antibody, the antigen, as well as the primary antibody attached with the marker. In here, the marker is combination of gold nanoparticle and magnetic nanoparticle, which will be indicated by the change of current if the interaction occurs.